We'll switch quickly from very deep quantum mechanical physics to biomedical engineering with our own Uri Nevo. Please.
Thank you very much. And yes, we are now like going up about 6 orders of magnitude gross of things. And this works relates to things we are doing in my lab. And I'll start with your request to just giving you a little overview of what we are doing. In my lab, the Laboratory of Cellular Biophysics and Imaging, we are located in biomedical engineering in the engineering department. We are doing things that are mostly on the interface of brain, biophysics, and MRI. Okay? And in some cases, we are doing things that are related to both three things. In other cases, just one of them. There is another issue related to eco-immunity, something that I'll mention in a second. I won't mention anything about this in the overview because these three guys are doing the things that I'm going to talk about in the lecture. I'll just mention briefly 2 or 3 small project slides. One is our work on characterizing porous materials by NMR. We are interested in water displacement in tissues, and this drove us to study and to try to invent things that are related to characterizations of porous materials, since tissues are modeled as porous materials. And we are working on developing ways to quantify really porous materials with polydisperse sizes in a sense, okay? And in this case, you can see, for instance, the quantification of sizes. These are already in a phantom that we created where we knew the partial volumes, and we managed to quantify the pore sizes and the relative partial volumes. Another work that we are doing, again related to NMR and MRI, completely not related to this lecture, is the use of what's called the NMR mouse, a machine that was invented. We bought it from a group in Germany, and we're using this machine in order to attempt to push it towards biomedical applications. This machine is used, this NMR scanner is used mostly for non-destructive testing, for NMR of tissues as well, and we try to push it to accelerate the ability to do imaging by various techniques that are mentioned here. Currently, we are working mainly on the infrastructure, on the techniques themselves, but eventually we want to go into quantification of bone and of tissues, of skin. Last thing that we are doing, which is completely irrelevant to nano or to biophysics, but is something that I like a lot and is related to things that I did during my postdoc, is something related to immunology. Again, it's the thinking here is physical, or the modeling is mathematical, but other than that, it's just biology. And this is an attempt to quantify the problem of biological problem of self-tolerance, how the body tolerates itself in a cell, how the immune system tolerates itself, tolerates the body without the need for various ad hoc laws. This is something that I can talk about for an hour, but not now. And let's go to the lecture. So the thing I'm going to talk about is related to things that happen within cells. But I'll begin with mentioning the concept of log driving. This is something that was done like 150 years ago, like a little before the invention of trains. And when you cut a forest and you want to use the trees in a distant place, the easiest thing to do in order to transport the logs is simply put them in a river. And this is a minimal investment of energy. You're simply using the flowing river. It fits very well rivers that are slow. It's reliable. I mean, everything eventually flows, although very slow. Sometimes there are jams over there. People needed to, those people are the log drivers that need to move the logs so there won't be jams. But the idea here is that you use energy that already exists there, simply gravitation, in order to perform transport. And we ask the question, and I'll in a second get to the reason why we ask this question, do neurons use such a mechanism in order to create transport, intracellular transport? I'll start with the incentive or the motivation for this study. We're working a lot in MRI, and specifically with diffusion weighted imaging. And with diffusion weighted imaging, the main observation that was discovered like 20 years ago was that when you perform a measurement of the diffusion or the displacement of water molecules in the brain, the displacement drops significantly post-trauma or post-stroke, as in this case. Do we have a... That's the wrong one. That's the wrong one. You have one, right?
Will you need the laser?
Just the
function?
laser. Just the laser
Then
function?
it should work. Yeah.
Okay.
Yeah. So it's the upper
Yeah.
part.
Thank you. So what you see in this image is a case of stroke. And in the look at the middle row. What you see here is the acute phase of a slice in the brain. And you see here hyperintensity in the MRI of that patient with an acute stroke. And that area is an area that was affected by the stroke. See, in a T2 weighted image, for those of you not acquainted with NMR, this is the main mechanism for decay of an NMR signal. We hardly see any effect here, although radiologists will be able to see something in this image. But the effect of the stroke is mainly seen in what we call the apparent diffusion coefficient, the measure for the degree of displacement of water molecules. And you see here the acute phase 24 hours afterwards, 7 days afterwards, and then 30 days after the stroke. What you see, the hyperintensity gives us an indication of a degree of displacement. The more displacement we have, the lower is the intensity here. And we can quantify exactly the apparent diffusion coefficient. We know that it's not like diffusion in a water bottle. And with time, we see a lower and lower degree of displacement. By 30 days, we get the opposite effect because we already have necrosis of tissues, and we have something which is more like free fluid inside the brain. So the question is, what's the cause for this? And this is maybe one of the main questions that we study in my group, since we don't fully understand it. For instance, our biophysical models usually treat the tissue as a porous material with intracellular fluid and extracellular fluid. And the intracellular fluid is usually regarded as the fluid that moves slower. But this work I mentioned here and the other works show that there is no good reason to assume this model. And this model is the basis for all the results that I mentioned here and other results. For instance, our view, and this is another case of a brain with the area affected by stroke in this case, our main view of the reason for dropping the displacement is that the tissue swells. This is something that we know that happens. Okay, the tissue swells, and instead of occupying something like 85% of the total volume, it occupies something like 97%. And then, if indeed water inside the tissue diffuses slower, then the average or the weighted average of the two volumes will drop down. But if you're taking the values, even the most extreme values of those that believe that we have differences in diffusion, and you put them in this model, you never get to the drop of 50 to 70% in the degree of displacement. Another thing is that water in the brain or fluids in the brain are not diffusing isotropically. When we have structures that are anisotropic, like white matter tracts, take for instance the two hemispheres in the brain, in between them we have like a parallel bus. This is the corpus callosum, just an ensemble of millions of fibers going from one direction to another. If I quantify diffusion by NMR in different directions, I will see that the diffusion along the fibers, this is like an axon of a neuron and sheathed with myelin. I'll see that the diffusion along the fiber is much faster than diffusion transverse to the direction of the fiber. And this can be easily understood if you take it that the reason for this anisotropy is simply the restriction posed by the membrane and by the myelin sheath. So one of the problems is that when we are taking the models that I mentioned before, or looking at the results, in many cases the diffusion, in this case we are not talking about a scalar, but a diffusion tensor, that when we diagonalize this tensor, the primary value of diffusion, the diffusion along the fibers, drops the most. So think of a tube that swells, and the diffusion that is affected the most, the diffusion along the fibers. And this is something that cannot be understood. And last issue that is related to poor biophysical understanding is the fact that maybe the apparent diffusion coefficient, the displacement of water molecules, oops, what did I do? Is related also to function. And here I mentioned work of Denis Lebihan from France, did these works in France and in Japan, and he performed fMRI measurement. This is bold fMRI, measurement based on blood oxygenation in blood capillaries and blood vessels. And then he performed a diffusion fMRI. And like in fMRI, what we're doing is simply stimulating some stimulus to the patient sitting, lying inside the MRI, and we are then detecting some activity in the region of the brain. In this case, there was a visual activity, and we detect something in the occipital lobe, the area in charge of visual perception. And what you see here is indeed this area was affected by the stimulus. This is the bold signal, but also the diffusion changed. Okay, so the Lebihan sees like an effect of 1 to 2%, a small drop in the diffusion coefficients. This was something really very controversial. After we published it, other groups published the fact that maybe artifacts related to the bold signal that pops out create this measurement, and we try to address this thing. I won't get to this issue because it will take you too long, but there are other measurements that also indicate that we have a problem. So one work that we try to do is first to try to quantify, do we have really a relation between brain activity and water displacement? And Nitzanti Roche, then a master student, took a model developed in the Hebrew University by Aroni Levtov. What she did is taking a spinal cord of a newborn rat. The use of a newborn rat is simply that we can excise it. The first thing we do is sacrifice the animal, we excise it, we take it out, and then we put it in a chamber and continuously perfuse it with artificial cerebrospinal fluid, the fluid that exists or that neurons like to see in a sense, perfuse it with this fluid with oxygen. And then we can keep the tissue vital for like 20 hours. Vital in the sense that if we activate it, we see action potentials, the firing of neurons. And what we did was we took this excised tissue and then we put it inside the MRI. Then we started to measure both MRI and electrophysiology. Electrophysiology, in order to detect activity, we are not at the moment able to do it synchronously, the MRI and the electrophysiology, but we did it outside. The measurement of electrophysiology were done with the help of a guy over there. And then we detect, for instance, the baseline period, the baseline activity of the neurons, and of the spinal cord. It's not only neurons. And then we apply a cocktail of excitatory molecules, serotonin, NMDA, and bicuculin. And we see something which is like an epileptic activity, very chaotic, very, very strong activity. And then we flush them out. And after hours, look again at the baseline activity. We see activity, but to a much lesser extent. And then we are doing a measurement of the diffusion weighted coefficient. And what you see in this image is the bath, the chamber with the fluid. Red is the fluid that is flowing inside the wall, the screen. And this is a spinal cord. The cross-section, I try to show you something on my screen. It's nice. An axial cross-section of the spinal cord, okay? And we see here, for instance, this is a map. This is not an image. This is the synthesized map of the apparent diffusion coefficient within each pixel. And you see, for instance, here, the values at the baseline level, the same values at the hyperactivity level, and then the return to the baseline level. And you see that the values of diffusion, as you see them here as well, during the hyperactivity phase are much lower relative to the values that we get in baseline activity. And this was done in various directions, in various diffusion times. We open a diffusion window for the molecules to diffuse in of 15, 60, 150 milliseconds, and we see the same effect. Our hypothesis for these results, so the issue is that we have a drop in the apparent diffusion coefficient, a drop in the displacement of water molecules. On one part of it is when we have trauma in the tissue, and secondly, when we have strong activity within the tissue. And our assumption is that we are not measuring only diffusion when we are using the technique of diffusion weighted imaging inside the MRI. We are measuring various mechanisms of microstreaming that occur within neurons. or within other cells. And we want to detect these eventually. And these mechanisms could be related, for instance, to flow. There are aquaporins within the membrane with water flowing across them. Maybe these are related also to the things that we measure. When we measure things in the MRI, where there is some diffusion window, and unless things are really synchronized, this is a pseudo-random thing. Things are not necessarily synchronized. We have swelling. Axons swell during the activity of the electrical activity. Maybe this thing creates something. There is electrical polarization. Maybe it creates something like electroosmosis. And I will talk about here about the issue of transport. Each neuron has inside it like a mesh, a network of various fibers, intermediate fibers, microtubules, actin filaments, and all of them carry on top of them molecular motors, simply molecules that carry with them vesicles or various organelles inside the tissue. So the question that we ask is, for Reynolds, all the time. So the question that we want to ask is this transport, that is something which is activated by the tissue, it's not something which is like passive diffusion, creates some drag in it, drag which is significant and we can detect it. So we are living in the regime of low Reynolds numbers, and I urge you, just write life at low Reynolds numbers and read a lecture, a very nice lecture by Purcell that got his Nobel Prize for NMR, but did very nice things in other fields as well, in which you can clearly understand that our intuition of life at low Reynolds numbers is completely poor. When I'm mentioning microstreaming, and we all think of, okay, of something flowing like in a very small channel, this is completely not the case. Okay? For instance, just to give you something that will really put me within the nano regime. Okay, this is one of two things that will really related to things that are sub-microns. For instance, if you are looking at an organelle that is being moved, or let's take a microbe that is moving inside some cell or inside a medium, and a microbe is the size of like 1 micron. Okay, it has a flagella. It has those tails that are revolving and creating this movement. And let's assume that at a certain point, the microbe, like a guy swimming in a pool, wants to enjoy the water for a second, just stops to move its flagellum. How long will be the distance that it will pass? It will stop at about a part of a nanometer. It will stop after a nanosecond. So there is no momentum here. There's no inertia. Everything is just the force that you apply at that moment, which creates some velocity, and that's it. When you stop applying the force, you immediately have blockage of any displacement. View it, if you want to use the Reynolds numbers, the relative Reynolds number for our sizes is that if you will swim inside a pool that is filled with tar, and you will move, each stroke will take like 3 weeks. Okay? This gives you an indication, the Reynolds numbers, or the biophysical case that we are talking about. So what Matan and Karen did was trying to use Stokes' equation. And once the guy in the audience showed me how they look much happier than Stokes. Although we don't get to, very small fraction of what so happiness is not related to science. Never mind. So they tried to quantify how strong is the resulting intracellular flow when we have movement of organelles, movement of vesicles inside an axon. So they modeled, as physicists, they modeled the cytoplasm as a viscous Newton fluid with very low Reynolds numbers. The organelles and axon are rigids. They are not, their membranes for these sakes are not moving. The organelles are assumed to move at a constant velocity. Again, something which is doesn't, we don't miss anything by taking this simpler approach. And we are using the Lagrangian specification, and we're taking the frame of reference of the fluid itself. What we want to know is what happens within the fluid. What is the distance that water molecules will do? So the first thing they did, was modeling the displacement of water molecules in blue when an organelle moves across them. Okay? And what you see here is the distance in micrometers when a single organelle is moving. the velocity of the moving organelle, by the way, is completely irrelevant. Okay? This is a low Reynolds numbers. If you're in that regime, it's completely irrelevant. And what you see here is the distance, the total distance traveled by different molecules. This is, for instance, the center of the spot where the organelle was moving, and this is the distance away from it in microns. Okay? But this is the only analytical thing that we are able to do when we are taking the case of free fluid, no restrictions, no nothing. And now we're moving to a closed tube. In a second, if somebody asks, are axons closed tubes or not? I'll address it in a second. Oh, I'll address now. We don't know. And so what we have, for instance, this is the center of the axon, and this is the wall of the axon. Okay? Again, this is the distance in micrometers. And what you see here is the total distance traveled by water molecules due to the movement of an organelle in this region. And what you see here is, again, flow created, and then reverse streaming that is caused because of preservation of mass, since we have a closed tube. And if we are doing just simply the integral of this, we get these distances. And what Matan and Karen did was quantifying this for various sizes, for various types of geometries. I won't get to it. And they saw a very nice law that the intersection is always the same here. And very interesting thing is that for a closed tube, you get a very short range effect. Okay? For an open tube, for instance, if our tube is open such that the pressures in all ends of the tubes are the same or are equal to the pressure in the surrounding fluid, then what you get is that a single organelle that is moving creates a flow in the entire tube. Much slower flow, but the entire tube has flow created in it. And this is, for instance, what you see here is a measurement of organelles moving. Okay, what you see here are organelles that were antibodies with some fluorescent molecules were attached to, and then they were tracked. And you see a time, and across time you see the distances or the path traveled by different organelles. And then what Matan and Karen did was trying to see, okay, let's take those curves that we got here, okay, and let's try to see if we get similar things. And indeed, let's take this model of an axon. In green here, you see water molecules at time 0. And then you have 3 organelles moving. First organelle moving from this point here, second from this point here, and the third one over there. And I'll now show you the path taken by 2 water molecules marked in black over here. So this is the first one. And you see, for instance, that the only thing that matters is how close the water molecule to the moving organelle. There is a decay length, of course, which is of the order of the organelles themselves. And afterwards, you get something that is hardly affecting. So for instance, for this water molecule, when organelle number 1 is moving along this track, along this microtubular actin, and is close to this water molecule, you get a very strong motion, very strong displacement. And when number 2 is moving, you get a slower one, and so on and so on. This thing is maybe not completely new, because in plants, you have the same thing. What you see here, a movie made for us by Menachem Moshe Leon from Hebrew University, is plant cells. And you see that there is a constant flow of water or fluid within the plant cells. The constant flow is created, again, by molecular motors. In the case of plants, they are moving on the walls of the plant. And they are creating this continuous flow within the plant cells. So what we are saying, maybe this mechanism is used also by neurons. Maybe the entire idea is simply that our neurons, in order to deliver, now let me just maybe say one thing. When we are talking about the transport of molecules within neurons, diffusion is completely irrelevant when we are talking about length scales of axons. I have axons that are of the order of tens of centimeters, those that are going from the top of my head down my spinal cord to the end to my bottom. And then those axons, to pass some molecule by diffusion, will take 30 years. Okay? But if I'm talking about transport, although this is a very slow transport, this could be a very useful mechanism. Maybe, we're speculating, maybe the non-understood mechanism of slow transport. There is fast axonal transport, which is known very good to be mediated exactly by, or not mediated, created by the molecular motors that are moving. Maybe the slow transport is in fact, or maybe part of it, a component of it, is really a fluid that carries various things that weren't encapsulated within vesicles. We have very problematic open questions that we are now trying to address experimentally. The first, are microtubules arranged in unidirectional clusters? 5 minutes, okay. In unidirectional clusters, if we have them arranged like this way, you know that molecular motors are moving in the same directions when they are close one to the other, then we'll have really a cooperative type of displacement. But if it's the opposite case, then if two nearby organelles will cancel one another's effect. Another question is the diffusion. Isn't the diffusion of the water molecules that shift them from one point to the other within the axon again cancel this effect? And the basic hydrodynamic properties of the cytosol, the fluid within the axon, are completely unknown. Okay, we don't know if it's open or closed. We don't know even the exact viscosity of it is not very well understood. So I'll now just go briefly through 3 projects we are trying to do now to use experimentally in order to quantify what we just suggested here. The first is to try to do this thing by NMR. Usually we are doing by NMR simply measurement of the apparent diffusion coefficient. We are performing measurements with different diffusion times and then try to look at the way the Gaussian of diffusion, is it a Gaussian? So we are measuring a propagator here. And we are trying to look if the propagator gets wider and wider, gets wider as the square root of the time law. If it's so, then it's really diffusion. But if it goes linearly with time, then probably there is some component of flow. We don't get conclusive results yet because of noise. We get differences between baseline and between excited states, excited neurons. But we can't say anything about it. Although I'd be happy to say now, but I don't have it yet. And the second thing that we are doing is attempting to use pulse sequences in MRI that won't detect only diffusion, but that will discriminate in between diffusion and microstreaming. Since in cases of microstreaming, when you have molecules that are moving constantly, you can cancel the attenuation caused by it. And this is something we're trying to do. And last thing that I'll mention that we're trying to do is the work we're doing with Uri Nevo from Bar-Ilan University. And we are using the model of the nervous system of the leech. Okay? And the issue with this leech is that we have very well ganglia. Okay? And each ganglion is composed with multiple cells, multiple neurons that we know to detect their exact spots. We are trying to inject quantum dots into them and then to track the motion of the quantum dots. Okay? This is something that measurement doesn't work, never mind. That was done over simply quantum dots inside the gel. The idea here is to use the quantum dots as a marker, which is like a colloid inside a fluid, and then to try to detect the motion of single quantum dots. We are not able to do it yet. We stopped this for a year. We are now resuming it. And our main problems are related to lots of scattering. We are not able to see real single quantum dots within the tissue. We don't know for sure that the quantum dots are not clustering together to form aggregates of dots and are not attaching to various proteins, although we are using quantum dots that are not supposed to attach. So lots of experimental problems that we are trying to address. And I won't get to the results, just of this intermediate results of these measurements. So to summarize, we use diffusion weighted MRI, microscopy and biophysical modeling to study the displacement of water molecules. We suggest that microstreaming is a significant thing that occurs within neurons. Okay? And if this is correct, then it might serve as an additional mechanism for transport within neurons. The transport is not only performed by is not only of what is really carried or attached to the molecular motors, but also to everything that is dragged with it. And this, if this is correct, then this should be also affected by neuronal function. I won't get now into the details of why it happens. And maybe it may aid us in resolving problems related to our understanding of diffusion tensor imaging. The students that did this work were Matan, Nitzan, and Keren. Eddie and Andrea are doing this work these days, and these are the guys we collaborate with. And these are finding agencies, and thank you.
So I thought I understood what it meant to develop a model.
Okay.
Apparently I never did. We have short time for some quick questions. Yes, Uri?
Hi. So two questions or remarks. One, you said that you don't know if neurons is an open or closed tube, but before that you mentioned something about
aquaporins, for example.
Yeah, but you mentioned something about the fact that the cells swell. Doesn't that mean that they are, in a sense, open to that level? And the second question is regarding the microstreaming. If you're saying that this is just because of organelles that are traveling and dragging waters with it, doesn't it will accommodate with the expressions of more motor proteins? Is that something
Expression of what?
Of motor proteins that will be doing the traveling, right? So does that known from the literature, whether after trauma or something like that, there is expressions of more motor proteins in the brain?
So first, with relation to the first question, it might be that what you're saying is correct, that the swelling indicates you're saying that there is entrance of water. But another model says that what happens there, and this is something we are also trying to ask by working on gels, is that exactly the mesh of fibers there is not something which is static. With different levels, for instance, of ions, this mesh opens or closes. And the measurement or the work on swelling of axons, the first guy who detected this swelling, his main suggestion was not that there's water flushing in from the outside to the inside, but rather that the entire mesh of what we call the ectoplasma, the cortex, the inner side of the cell, is changing, and this is what causes the swelling. With respect to the second question, we ask about cases of trauma where you have different expression of proteins. First, I don't know, but I know that within trauma you have so many effects that occur that I wouldn't know how to separate the effect of what is expressed more or less and then affects this. If I understood correctly what you're asking. You can, okay.
So you mentioned that the elasticity or the properties of the axons are unknown, but there are a couple of papers where they basically did experimentation and then developed a model where they used a bent needle, and these.
So one of the things is again that, and we suspect this is something we're trying, and it's related to what I answered, that this elasticity changes, and it changes, of course, when we have trauma, but it changes also in cases of electrical activity. Okay, when I have electrical activity, the entire mesh may be changing, and I don't know it. I don't know the exact, I don't know the exact degree that it happens. Yeah, this is my answer.